Hello, everyone. Thanks for joining us for the Forced Degradation of Proteins Through Mechanical Stress webinar. My name is Matt Willard, and I'm the Senior Marketing Manager at Fluence Analytics. Before we start, please note the Q&A and chat features at the bottom of your screens. You can use these to pose questions or make comments during the webinar, and Dr. Duran will respond to them at the end. Fluence Analytics is a startup headquartered in New Orleans, Louisiana, that spun out of Tulane University in 2013. We provide analytical products which yield continuous data streams for industrial and lab applications in the chemicals and pharmaceuticals industries. Earlier this year, Stephanie McCormick, the ARGEN product manager, asked Dr. Duran to share his work about using ARGEN to determine the impact of mechanical and physical stress on protein stability. After the release of the technical note, we received many questions on how ARGEN can be used for shear and stir stressing and with various pumps. Today's webinar will address many of those questions, and it will also discuss benefits from applying mechanical stress and material interactions while collecting kinetic aggregation data. Our panelist is Dr. Kurt Duran, a research scientist at Tulane University's poly-RMC. Prior to his current position, Dr. Duran worked as a senior instrumentation specialist at poly-RMC, where he often used ARGEN for experiments and applications. And with that, I'll turn the webinar over to Dr. Duran.
All right, thanks, Matt. So as Matt said, what we're going to be talking about today is the forced degradation of proteins through mechanical stress. The general purpose of this talk is to look at expanding some of the capabilities of routine ARGEN analysis to look at other types of stir and mechanical stress on proteins and how we go about setting up those systems for analysis. In addition to that, I'm going to have a bit of detail on coupling the ARGEN system to secondary detector systems to expand the capability of the system. So with that, let's kind of briefly introduce the ARGEN system. I know that most people here are probably familiar with it already since you're at the webinar, but the ARGEN itself is a system that's capable of continuously monitoring light scattering from 16 independently controlled cells. We have independent control of both temperature and stirring for each of the cells. And one advantage of this independent control is the samples can be started and stopped independent of each other. So you can basically be taking things in and out and running them continuously. We're going to sort of harness that capability for looking at coupling to basically secondary systems or expanding capability through different types of stirring or peristaltic and shear stress, and using the ARGEN to monitor that aggregation. So the system itself is capable of early detection of aggregation and degradation of proteins. That's what a lot of people use it as its main purpose, but there's a lot of other applications you can get out of the ARGEN system. The key outputs that are usually taken from this is the aggregation rate, which is basically just the change in the normalized molecular weight of proteins versus time in terms of reciprocal seconds. And that can be used to determine an absolute value for the aggregation. Also, you can get the normalized molecular weight as the system changes through either thermal or stir stress in the system. So a number of applications for this would be formulation stability of proteins under both thermal and stir stress, interfacial studies, material interactions. It can be used for scheduling for GPC injections, looking at the degree of aggregation and then scheduling that for GPC analysis. It can also be used for things like dissolution kinetics of samples. And then there's a host of other things, and a lot of those I'm going to go over today during the course of the talk. So just sort of a little cartoon outline of protein aggregation. What we're looking at in terms of aggregation, the system is taking a native protein, applying some stressor to induce a partial unfolding of the protein, and then you have aggregation, which occurs by diffusion-controlled mechanisms. Basically, the proteins clump together, and then that causes an increase in the light scattering signal, which we can monitor and then correlate back to the change in molecular weight or determine aggregation rates. Protein aggregation, the reason why people are primarily interested in studying this is that it's a pretty big problem in development of new therapeutic proteins. If you have aggregation of the material, it's going to occur through either storage conditions, transfer, basically even going through a syringe can cause shear stress on these. If you have aggregates form, it can render the protein either ineffective or in some cases even induce an immunogenic response. The mechanism for aggregation, there's numerous mechanisms which can occur, and it's highly protein dependent. So because of these problems, the ARGEN is sort of ideally suited in that it's good for a high throughput screening. You can simultaneously run up to 16 samples at once. You can quickly cycle through them. So it's a great way of doing an initial characterization of stability. So as I mentioned, the routine application for ARGEN, and this is the stuff that's built into the software, it's capable of easily generating this out, and that is looking at thermally induced aggregation. So taking a sample, holding it at isothermal condition, monitoring the change in the light scattering signal. We find that for thermally induced aggregation, the aggregation rates that are determined are highly reproducible. Generally speaking, we have relative standard deviations of, in the order of a couple of percent for reproducibility. In contrast, when we look at contact stir-induced aggregation, by contact stir, I mean a PTFE stir bar sitting at the bottom of the cuvette. And that is typically how most people would do their stir stress on an ARGEN. When we look at that, what we find is that there's a wide variation in the aggregation rates. We can have RSDs of up to 15, 20% or more. It's basically, it's not terribly reproducible. So that got us curious if we could look at other ways to do a stir stress and see if we can, basically increase that reproducibility or see if there's some differences in between contact and non-contact stir stress. So some of the examples I'm going to go over for some of the non-routine methods today, as I mentioned, non-contact stirring. The first attempt we look at this was using an overhead suspended magnetic stir bar. That allows us to control the stirring via the ARGEN software. Basically, we couple it to the stepper motor in the ARGEN and then control the stirring through that. To construct those cells, it's actually fairly easy to do. It can either be done through a 3D printed construction, or if you have some simple machining tools, it can be done that way. And then for actually making the impeller for the stirring, those items are easily obtained also. It's a matter of just having either some stainless steel or PTFE rods and some bearings to put them through. So it's not a big deal to do. We've also looked at non-contact stirring using an overhead impeller controlled by a stepper motor. Again, that's fairly easy to construct and put together. The reason why you'd want to consider using a stepper motor as opposed to magnetic stirring is that the stepper motors are capable of going to a higher stir rate. And that has to do with some specifics on how the magnetic stir bar is set up in the ARGEN. I'll go over that in a few slides. We've also looked at capillary shear stress through different tubings and tubing diameters. We've done that by using a reciprocating syringe pump to recirculate sample in and out of the ARGEN cell while doing monitoring in real time. Other examples of things we've looked at is recirculation of proteins via peristaltic pump. When we had originally looked at that, we had proposed using a peristaltic pump as a method to introduce samples to a secondary detector. And what we found is that even though a peristaltic pump is considered a very typically gentle way of recirculating materials, we actually did see aggregation. So we sort of invested, we kind of investigated that further. I'll go into some details on that. In addition to the peristaltic recirculation, we've coupled syringe filters to look at filtration effects on proteins via peristaltic recirculation. And then I'm going to have a number of slides on looking at continuous extraction of samples from the ARGEN cell and going through a secondary detector. That can either be a multi-angle light scattering system, fluorescence, UV, etc. And that's done via syringe pump. And I'll go into the schematics on how we set that up. And then I'm also going to cover a little bit on using either continuous or discrete extraction of materials for GPC analysis. I know some people already do GPC scheduling. This is simply another way to sort of pull the sample from the cell And I'll kind of go over that as well. And a few other items I'm going to talk about just briefly at the end is looking at doing direct titrations, either salt excipients, acid bases, etc. That can be done in the cell in the ARGEN system. Look at generation of Debye plots for calculation of virial coefficients using the ARGEN. And then lastly, I have just a quick slide on looking at sort of some novel stuff we did, which is looking at supramolecular host-guest interactions using ARGEN. So for setting up these experiments, they're generally pretty simple to get going. As I said earlier, for setting up the non-contact stirring, either using a 3D printer or some basic machining equipment to construct the cell cap, as well as setting up things for the spindle and the bearings. So it's pretty straightforward. We get a lot of our small parts from a place called McMaster Car. They're a great source for little small parts and unusual things like that. Also, we've gotten stuff off of Amazon. Generally speaking, it's pretty inexpensive and not difficult to do. Another thing that you'd want to have is a syringe pump if you're looking at capillary shear or withdrawing through secondary detectors. As I mentioned, using peristaltic pumps for withdrawal can cause some problems due to just basic aggregation caused by the peristaltic pump itself. And then, as I said, with the peristaltic pump, we've used that for recirculation and filtration experiments. And then for the liquid transfer, just some HPLC tubing and small fittings. So all of this is very inexpensive, and generally everyone's going to have this in their lab already. One note on looking at some things such as doing the titrations and Debye plots. I just want to mention if you aren't familiar with using the data export function in the software, it's really a much easier way to handle a lot of these types of experiments. You can generate the file, export it out for external analysis in Excel or ARGEN or whatever else you want to use. So that's just something to familiarize yourself with if you plan to do these things. It's quite easy to use. So non-contact stirring. As I mentioned earlier, what we saw was a lot of irreproducibility in contact stirring, and we weren't sure of the mechanisms behind that. So we wanted to probe some other ways to look at stirring in there. And what you can see in the top center, on the left-hand side, there's an example of some of our machined caps. You can either use aluminum, I think these might be Delrin blocks that we used. Going through the middle of the block at the top, we have a series of 2 bearings in there with a central shaft, and then that's coupled to a magnetic stir bar at the bottom. Now, one thing to note about that is that when you're setting this up for doing analysis on the ARGEN, the optical path in the cell sits about 3 to 4 millimeters from the bottom of the cell. So that somewhat limits how far you can move that magnet down and still couple it to the ARGEN itself. And as I mentioned earlier, generally for high-speed applications, above 500 RPM, you're going to want to use something that's not using a magnetic stir because you tend to break that magnetic coupling if you go above that. On the right-hand side of there, we have just an example of a stepper motor set up with the cell. So we have the stepper motor set with the PTFE shaft. You can use whatever kind of impeller you want on there. Design that out yourself. Again, that's a good application for 3D printing to develop those. And then on the left side, I have just a little quick video. This shows one of the magnetically coupled stirrers in the ARGEN itself. So you can see that that spindle is spinning there once we kick up the stepper motor on there. So when we look at the differences between overhead versus contact stirring, what we see is that we get a markedly decreased aggregation rate with non-contact stirring. And in addition to that, we've noticed that in what we've examined so far, at least, that the reproducibility increases pretty drastically when we look at non-contact. And we also see something that's kind of unusual. We see a lag phase. It's pretty common in a lot of proteins when we look at this. And that can go on for hours to even a day or more before we start to see aggregation. So if we look on the plot at the right, you can see in pink, green, and black is a typical contact stirring, and these are all run at the same rate. So I'll run at 50 RPM. And then in the red and blue at the bottom, that's our non-contact overhead stirring using a coupled magnetic stir bar. So for examining capillary shear stress, again, this is pretty simple to set up. What we use is a reciprocating syringe pump with a given length of tubing, whatever material we're interested in looking at. On the picture of the right is an experiment in which we looked at comparing a 1 meter length of PTFE tubing versus stainless steel versus PEEK HPLC tubing. A couple of different diameters for both the PEEK and the PTFE. Doing these experiments, if you want to do a direct comparison, it's important that the same length of tubing is used, obviously. You also want to have an equal starting volume for the samples and consistent flow rate for direct comparisons. Now, what we found in our initial studies is that kind of surprisingly, since it's capillary shear, is that the diameter of the tubing actually has less of an impact on the aggregation rate that's observed than the material we're using. And for the initial system of 5 different tubings that we looked at, we found that aggregation is highest in PTFE tubing, followed by stainless steel and then PEEK. And then in the plot at the bottom right, you can see where we have that aggregation rate versus time. On the bottom, red is a control sample, which isn't being recirculated. So there's a fairly significant amount of aggregation that occurs over the course of a day or two here. Another thing that we've looked at is halting either the stir or the shear stress to examine the effect. And sort of intuitively, you would think that once the stress is stopped, that aggregation would stop. And that's what we found, not surprisingly. So on the right at the bottom, we have an example of when we were looking at a reciprocating capillary shear via syringe pump, and we had a syringe failure that occurred during the experiment. And you can see that when that failure occurs, our aggregation halts. We get a linear response to the light scattering signal. On the left, we've looked at applying stir stress. This is contact stir stress, which is started and then stopped at periodic intervals. And what we see is that when the stress is stopped, our light scattering signal basically flatlines. And when we start the stirring again, the aggregation profile continues along the same pathway as it did before. So you can basically just halt and start the aggregation during these experiments. So I mentioned earlier that we had looked at peristaltic recirculation as a method for introducing samples to a secondary detector. And what we found, to our surprise, is that for a number of proteins, we see some fairly rapid aggregation occurring during peristaltic recirculation. On the plot at the right, you can see for BSA, which is in red, the aggregation occurs so quickly that it's almost hard to put it on the same scale as some of the other materials studied. It quickly goes to a normalized molecular weight of 12 before we go through an ND filter switch. And that happens within a matter of, I think, less than 10 to 15 minutes. So it's very unstable under peristaltic recirculation. The way we set this up in the sample cell is we basically take a Teflon cap, drill a 1/16 hole through each side, and then insert some HPLC tubing. And when we do this, we have one set of the tubes that goes all the way to the bottom of the cell, others withdrawing from the top. That way we get a bit of a flow through the cell. So it's sort of just a little bit of a homemade flow cell that we've set up there. But we've gotten some pretty interesting data out of that. We found for a number of proteins, though, that under peristaltic recirculation, they're quite stable. And it's not terribly surprising, since we've seen a wide variation in stability to other types of mechanical stress. And on that plot at the right, you can see that we've also run some controls on there. We have a PBS buffer as well as a water control. We were initially concerned that what we might be seeing in the light scattering might be shedding of the particles from the peristaltic tubing. So we wanted to just double check that. And in fact, we found that just recirculating without the proteins in there, we don't see any evidence of an increase in the light scattering signal. So building on the work with the peristaltic recirculation, we've looked at doing that with different syringe filters in place. We've connected them within the recirculation loop via Luer lock fittings, so they're easy to switch in and out. On the particular plots that I'm showing here, what we've done is look at recirculation on the middle plot at the bottom. In red, we have the 90-degree scattering signal from ARGEN for just a sample recirculated without filtration. And then in blue, we have one with I believe that's a 0.22 micron cellulose acetate filter in place. The plots at the right kind of explain these. We developed a detector in-house for looking at light scattering spikes. Basically, it's a 90-degree scattering detector, which uses a high-speed detection system on there. So we're able to sample at several hundred hertz frequencies or higher. And by doing that, you're able to actually see when these large aggregates pass through the optical path. They'll result in a spike in the scattering signal. And so what you end up with is a profile where you have these spikes from large aggregates occurring on top of your baseline light scattering signal. And on these plots at the right, these are shown from the first hour of the sampling and then the last hour of the sampling. The top plot is the protein recirculation without a filter in place. And what we see is a large number of scattering spikes initially, an increase in the average baseline light scattering signal by the end of the experiment, and the spike counts have really not changed much. We're still seeing a lot of these large aggregates flowing through. On the bottom, we're doing the same type of experiment with the filter in place. And again, we see initially a large degree or a large number of these large aggregate spikes occurring. And as we get to the end, we've seen really no increase in the baseline light scattering signal and also a decrease in the number of spikes we're seeing go through there. So the filtration is removing these large aggregates that are forming during the peristaltic recirculation. So next I want to look at coupling the ARGEN to a secondary detector. And this can be used for with a number of different things. Basically, the way we do this is we use a syringe pump at the end of the detector train to withdraw a sample through the detector, which is pulling directly from the ARGEN cell. And we couple these together just via HPLC tubing. For a secondary detector, examples would be a multi-angle light scattering, UV, fluorescence, etc. One thing to keep in mind when doing this is that the dead volume in your secondary detector needs to be fairly small. The ARGEN system itself, the cuvettes that are in there are capable of holding about 3.5 mils of total material. So that necessitates having a small dead volume in any secondary detection methods. Another consideration is that when you are pulling from the ARGEN through a secondary detector is you need to match your flow rates to your expected time of your experiment. So if you're going to be looking at something that's occurring over multiple hours, you want to go for a very low flow rate so that you don't basically drain out all of your sample. Another thing you can do besides looking at just the secondary detector system is you can couple this directly to an auto injector for GPC. So instead of pulling directly from the sample cell during a run, you can just have this automatically going through an auto injector, which you can set up for periodic injections. Something else to consider is that to get your baseline values, you want to begin pulling your sample through the system before you actually begin applying your stressor. Because of the low flow rates involved, oftentimes if you start your pumping at the same time as the stressor is applied, you're looking at anywhere from, it could be 30 minutes to an hour of delay time before you're starting to see sample. So just keep that in mind. And then obviously, due to that low flow rate, you want to minimize your dead volume in your transfer tubing from the ARGEN to your secondary detector system. So for an initial example of some work we've done using secondary detection system, this is looking at stir stress versus thermal stress via coupled GPC. This system was set up again with using a syringe pump to pull through the auto injector, and then we would periodically inject onto the GPC column. And we see some pretty drastic differences in the GPC data between the thermal and the stir stress. If you look on both the top left and the top right, these are the ARGEN generated plots of the aggregation profiles. On the left is the thermal stress. So it's a monoclonal antibody held at 70 degrees C. On the right is the same antibody. It's being stirred at 1,000 RPM via contact stir stress. And you can see that they both have a significant amount of aggregation occurring via the ARGEN signal. But when we look at the GPC data for thermal stress, what we see is on the bottom left, you see a decrease in the monomer peak, followed by an accumulation of the aggregate peak by the time we're sampling out at 1,000 seconds. With the contact stir stress, we don't see any evidence of aggregates in there. And in fact, the initial monomer signal doesn't even really change over the course of this. So mechanistically, there's something different occurring between these samples. So to probe that a little further, we expanded on this idea of using secondary detection to incorporate a multi-angle light scattering system into there. So what we now have is the same basic detector train pulling with the syringe pump to the GPC, but we have a Malls detector also in line on the GPC. And the reason for doing that is that for the Malls, we can look at the angular dependence of the proteins and determine basically if we have either a large population of small aggregates or a small population of large aggregates. Remember, for light scattering, the signal you're seeing is simply an average of all the aggregates that are in your sample population. So a small population of large aggregates can give the same signal as a big population of small aggregates once you average everything out. With the Malls detector, we're able to differentiate between those two. So, as I said earlier, with the small population of large aggregates, we end up with a strong angular dependence in the signal. So we looked at BSA under contact stirring. And again, we're pulling, if you look on the top right plot, this is the Malls data at various time points throughout the stirring process. So our red is our T equals 0, baseline sample. And then as we go through up to 3,500 seconds, we begin to see a strong angular dependence that's occurring. When we look at the GPC data on the bottom right, as in the previous slide, again, we don't see any change in that monomer signal. So if you're looking at this as via GPC, you're not really seeing anything occurring. It looks like you're not seeing any aggregation at all. But when we look at the thermal stress, using the same setup where we're pulling at periodic intervals. Again, this is BSA. I believe it's being stressed at, I think, 58 degrees C. So in contrast to the stir stress, we see very little change in the angular dependence. If you look at that plot on the top right, even though we're getting and I have the light scattering data from the ARGEN on the next slide. I can show you that. But we have a strong increase in the light scattering, but very little change in angular dependence. And when we look at this by GPC, we looked at this on an aggregate-specific column. I believe it's a TOSO ultra aggregate SW column. It's capable of actually separating out dimers, trimers, etc. And when we look at that over the course of the run, what we see is on the bottom right, we see a buildup of those aggregate peaks occurring. And you can almost follow this through that formation, the dimeric, trimeric, etc., aggregate formation as we go through the experiment. And we also see a corresponding decrease in your monomeric peak over the course of this. So if we look at this in just a little more depth here, so this is showing the ARGEN data as well. If you look on the top right, so again, it's thermally stressed sample. We're pulling periodically from that. Well, we're continuously pulling from the ARGEN cell, but periodically injecting with the autoinjector onto the GPC. And again, just showing the sample here. This is over the course on the bottom right of 0 to 286 minutes. You can see the loss of that monomeric peak with the buildup of aggregates there. And again, just to reiterate this, under the contact stir mechanical stress, we don't see that same process occurring. We just simply don't see these dimers, trimers forming. On the converse side of looking at the formation of the aggregates, you can also monitor the protein monomer loss by coupled GPC. And that's basically just normalizing the peak area of a given sample to your starting value. And you can plot that out to determine the monomer loss. And that's, an application, if you don't have an aggregate specific column, that's one thing that can be done. Simple procedure. So to dive a little bit deeper into the differences between thermal and stir stress, we hypothesized that if an aggregation process is mechanistically the same for a sample that's given under a given stressor, for this example, we're using thermal stress. We propose that if the mechanism is the same, then if they should be able to be superimposed upon each other by changing the time base of the sample. So when we look at that, we have a sample here to thermal stress from 78 to 84 degrees C. If we adjust that x-axis timeline and scale it, what we see is in fact that these samples are directly superimposable. In fact, there's almost, really can't see much daylight between the different samples. So that's a pretty strong evidence that mechanistically we're seeing the same process here. When we extend this out and look at the differences now from the ARGEN on the stir stress versus thermal stress, for the same sample, we have a sample that's stirred at 1,000 RPM in red and one that is thermally stressed at 70 degrees C in blue. We find that even though they're reaching roughly the same normalized molecular weight values, that they follow very different pathways on the way to that increase. So that's a pretty strong indication that we have different kinetic mechanisms there. And of course, that's been reinforced by our work with the mols with the GPC. So it appears that for thermal and mechanical stress, these are two mechanistically very different processes. For the last couple of things I want to go over, I talk about a few other things that are pretty simple to do with the ARGEN, but sometimes people don't think about. One is generation of Debye plots using the ARGEN. And that is fairly easy to do. Basically, how we do it in the lab is that we start with a concentrated sample that's at the upper end of the concentration range we want to probe. We'll put an aliquot of that into the ARGEN, generally as small a volume as we can get away with. For a 1 centimeter cell, the minimal volume is around 600 microliters of sample before you start to interfere with the optical pathway. So what we do with that is we take that concentrated sample and then just titrate with our buffer and run through serial dilution and generate this concentration dependence versus a light scattering signal. From there, we can go ahead and convert that into a Debye plot, normalizing with toluene, just kind of standard procedure, and then we can go ahead and calculate the virial coefficients for that. And this, like I said, this is a pretty easy thing to do. The only thing that is a little tricky with doing the Debye plots, and this is true whether you're doing it on ARGEN or not, is that your lab skills are pretty important. It's really easy to introduce bits of dust, contaminants into the cell. So it is a little tricky to learn at first, but once you get the method down, it's pretty quick to do these. Another thing that I'll note is that for a wide concentration range, if you look on the plot at the top, we're going from roughly 150 to 200 mg per mil down to essentially 0. To cover that range, when you go through the serial dilutions, at some point you will have to remove an aliquot from the cell. So what we'll do is we'll pull an aliquot and just continue diluting with our buffer from there. And it generally requires a few different times you'll have to pull that sample out to continue on with your path. Another thing you can do is at the start of the data collection is to go ahead and prepare samples at a few different concentrations. and you'll transfer those in at the given point, and continue on with the titration work. Another thing that we've looked at, which is kind of interesting, it's one of our collaborators here at Tulane. We looked at using the ARGEN for probing host-guest interactions for supramolecular systems. And basically what we're looking there for is a variation of the host-guest ratio. I'm looking for a signal maxima from the light scattering system. And it turns out that it actually works quite nicely. It's very easy to do. So it's a very, very simple way to do this compared to how this is sometimes often done. So that's another thing to think about. And we've had some thoughts of doing this, looking at, for example, like antigen-antibody interactions as well, using a similar methodology. So kind of move on to the summary and then questions here. So just as we mentioned, the ARGEN, it gives a simple and robust platform for doing protein aggregation by the typical methods, by thermal and contact stir stress. All of that's built into the system. It's very easy to use. Expanding that capability is quite easy. A small amount of equipment, a little bit of setup, and you can start looking at things, non-contact, shear, all of those things. And I'm sure there's other things that I didn't touch on in the talk that people can think of to do. So for the examples of what we've shown here that are fairly easy to set up, the non-contact stirring via magnetically coupled stir systems or using stepper motors, peristaltic recirculations, as well as looking at filtration effects via peristaltic recirculation, capillary shear via reciprocating syringe pumps, and then looking at secondary detection for monitoring formation loss of aggregates as well as mechanistics for that. And then lastly, as I mentioned, the generation Debye plots, titrations, and probing host-guest interactions. So with that, I'll kind of turn it over for any questions.
Thank you, Kurt. That was a great presentation. So if any of the guests have questions, you can use the Q&A feature at the bottom of your screen, or you can also just throw them into the chat function. I have a couple right now. In regard to slide 13, Kurt, have you ever seen filtration promote aggregates? Also, have you tested different filters with different materials?
In the studies we've done to date, I have not seen filtration promoting aggregate effects. Though that, theoretically certainly is possible through shear effects through the filter.
We have collaborators who have seen filtration produce aggregation. We have very limited studies.
Yeah, this Professor Wayne Reed is sitting in here with me. He's a director of polymer MC, and he was saying that we do have some collaborators that have seen aggregation through filtration effects. We personally in our lab have not. We have looked at different filter materials, but we really haven't compiled enough data to see effects on that.
Okay, thank you, Kurt. Regarding slide 11 in the peristaltic recirculation, protein C was stable and protein A aggregated. Was this different than other characterization data? Was it different from thermal stress?
Yeah, all three of those proteins showed different degrees of aggregation or thermal and contact stir stress.
Okay. We don't have any other questions right now, so if anyone wants to ask Kurt a question, please submit it. Otherwise, we'll just wrap up the webinar. Okay. Well, thank you again, Kurt. That was a very informative webinar, and I hope our guests really got something out of that. If anyone has follow-up questions, I'll send out the slides and the recording of the webinar either today or tomorrow. And if you have additional questions, feel free to respond with those. So I'd like to thank everyone for joining us and wish everyone a great afternoon. Thanks again, Kurt.
All right, thanks, Matt.